Good morning children, I am Sadhaisis. Now I am going to teach you the proof for DNA as the genetic material. And the experiment was conducted by two scientists, Alfred Hershey and Martha Chase. So the experiment is commonly known as Hershey and Chase experiment. They were conducted an experiment on bacteriophage and E. coli. So we can see bacteriophage. Bacteriophage means viruses which infect or attack bacteria. So the virus which can infect bacteria or the virus which can attack bacteria are commonly called bacteriophage. I will show you the picture. So here this is a bacteria which is attacked by a phage or a virus. So this is E. coli bacteria. E. coli Escherichia coli bacteria, and it is attacked by T2 phage. So T2 phage is a kind of bacteriophage that is commonly attack the Escherichia coli or E. coli. And the T2 phage has two structural components. They are DNA core and protein sheath. These are the two structural components of this T2 phage. Okay, so T2 phage is a kind of bacteriophage that can infect the bacteria E. coli. And the two structural components of the T2 phage are DNA core and protein sheath. So here we can see there are three steps in their experiment. The three steps were, first one, infection. That is the first step. Second step, blending. And the third step is centrifugation. These are the three steps in the experiments. And they were made two preparations. So let's see which are the preparations. So the two preparations, the first preparation, in the first preparation, the protein sheath was made radioactive with radioactive sulfur, that is S35. Okay, so in the first preparation, some bacteriophages were taken. The protein sheath of that bacteriophages were made radioactive with the radioactive sulfur. Okay, that is the first preparation of bacteriophage. First preparation of bacteriophage or that virus T2 phage. And in the second preparation of this bacteriophage or T2 phage was the DNA core. DNA core was made radioactive with radioactive phosphorus. These are the two preparations made by these scientists in their experiment. So in the first preparation, two preparations were made by the scientists. In the first preparation, the bacteriophages, the protein sheath of the bacteriophages made radioactive with the radioactive sulfur. And in the second preparation, taken some bacteriophages and those DNA core was made radioactive with the radioactive phosphorus. So these are the two preparations made with bacteriophage. So clear? And these two bacteriophages were allowed to infect. So these are the two preparations. So in this preparation, the DNA core is radioactive. Radioactive with the radioactive phosphorus. Okay. So here the protein part is radioactive. Protein sheath is radioactive with the radioactive sulfur. Okay, so here this yellow color indicates the DNA, radioactive DNA, and the blue color indicates the radioactive protein. Is it clear? And next we can see both were allowed to infect. So this is the yellow part, you can see this is the blue part. So here the radioactive phosphorus bacteriophage infection we can see. So this is the E. coli bacteria. E. coli bacteria here, E. coli bacteria. Okay. So first the radioactive phosphorus labeled, that means P32. So the phage or the virus with the radioactive phosphorus is allowed to infect the E. coli. In the second case, the phage with radioactive sulfur allowed to infect the E. coli. Next we can see, so as I told you, the second step is after infection, the preparation were blended. So agitated by using a blender to separate E. coli and phage. So after infection, the second step is agitation or agitated by using a blender. So this was done to separate E. coli and bacteriophage. So from the infected E. coli we can separate bacteriophage. We want to separate both. For that purpose there is an agitation by using a blender. Then it is centrifuged. That is the third step. Okay, after blending it is subjected to centrifugation. So after centrifugation, the bacteria as the heavier part, it settled down. So we can see the bacteria is settled down because it is heavier than phage. But the phage, you can denote the phage like this. So the phage remain as supernatant because it is lighter than bacteria. Okay. So this is the result after centrifugation. Okay. Now we can see what are the observations and conclusions in both the preparations after blending and centrifugation. So the three steps are over. First, infection. Second, blending. Both the preparations were blended. Okay. Then centrifugation. So after centrifugation we can see in the first preparation. So the observation and conclusion we are going to see here. The bacterial cells are here settled down as they are heavier, and here this bacteriophages, you can see it remains as supernatant as they are lighter than bacteria. And here in the second preparation also we will get like this. Here the bacteria settled down. Here the phage remain as supernatant. Okay. So the conclusion was here the phage and this is the bacteria. So the first preparation, as you know, protein coat was made active. Radioactive. Here this bacteria is not radioactive. Bacteria is not radioactive, but here the phage's protein part is radioactive. Protein part is radioactive. Okay, so after centrifugation, they observed that the phage which remained as supernatant, the protein part was radioactive, but the phage which is infected, or we can say the bacteria which were infected by this phage is not become radioactive. So the conclusion was the bacteria infected with virus. Which virus? T2 phage. Okay, T2 phage or T2 bacteriophage with radioactive protein were not radioactive. Is it clear? So actually this protein part were made radioactive by using sulfur. But after centrifugation, the protein part became radioactive, but the bacteria which were infected by the same phage whose protein part is radioactive do not become radioactive. That means the protein cannot transfer anything to bacteria. Is it clear? In the next preparation we can see here the DNA part was radioactive. Here the protein part was radioactive. Okay. Here they were observed that the DNA part here is radioactive. DNA is radioactive, and this radioactivity was made with the phosphorus, radioactive phosphorus, and the bacteria which were infected by the virus whose DNA part is radioactive, that become radioactive. Okay, so the conclusion was bacteria which was infected with virus with a radioactive DNA become radioactive. Okay, so here this proves that the DNA is the genetic material, and here the radioactivity is transferred into this bacteria. Okay, so this proves that DNA is the genetic material that can transfer characters from here to this bacteria. So here the protein part cannot transfer the character to the bacteria. So this experiment, Hershey Chase experiment, proved that DNA is the genetic material. Is it clear? Next we can see what are the characteristic features of a genetic material. Okay, so the genetic material must have the following characteristic features. A molecule should be, or it should be able to generate its replica during replication, because they are able to produce their own copies. It should chemically and structurally be stable. It should provide the scope for changes, mutation. Changes means mutation in the genetic constitution, change in the genetic constitution, that is mutation, that are required for evolution. It should be able to express itself in the form of Mendelian characters. So these are the basic characteristic features of a genetic material. Here the genetic material is DNA. Hope you understood everything about Hershey and Chase experiment. That's all for today. Thank you.